How do I help you with your stress? You're not stressed on this course, though, are you? A little bit? Why? Everyone.
Spring break.
Because of the exam.
The podcast, our presentation.
The side question.
But those were there before spring break.
We don't have time to do that.
Fair. All right. Well, do you want me to leave off the side learning questions for chapter 9?
Yes.
All right, they're gone.
I was going to agree with that.
Really? Okay. All right, let me see if I can pull up this PowerPoint. How to open up PowerPoint. Where are my files?
Okay, Don, I set a few.
Do you understand how I couldn't maybe on my screen? It was
I
just
had no idea.
you don't. Let me. Oh, no, I can't.
I thought my first drawing was pretty nifty.
Well, what does it look like on your screen? Because on my screen it just looked like red blurs and orange blurs. Oh, that's great. That is not what I saw at all.
Can you imagine if she called you over here and you saw like the red blurs again and pages
It's great.
This is beautiful.
It's clear. I saw this. I'm like, did she write our name? Because that would be a big difference.
Yeah.
This is all I
I sent this again, and then I sent a PDF.
Okay.
You can really
Either.
Right. They do that.
No, this is good. I thought maybe you were writing pictures, but I just couldn't tell.
It's a
So.
little blocks.
All right, that's good. All right. Let's present this. How do I hit presenting mode? Slideshow. From start. You have a 401k already?
Maybe.
That'd be impressive. Yeah. Mhm. Okay, so we're going to do the immune system today. Most of those who took 406 know a lot of these concepts. Did they feel, to the grad students, did it feel like you knew some of this? Okay, good. So it's not like brand new concepts at all. Fantastic, good. So then this will go quick. Except that I lost mine. All right, Sam. Morning, Sam. Yes, she's taking a drink. Tell me something about innate immunity. One thing. One word. Describe it. How would you tell somebody the difference between innate versus adaptive? Okay, so it doesn't evolve. That would be a correct thing. So it does not. I'll go with that. Adaptive, where it changes, and at some point we have this as part of the chapter, but it's the shuffling of genes to make antibodies. We don't shuffle all the time because we don't need all the antibodies, and sometimes you have to get exposed to something first. Then that makes the antibody. So when you get exposed to something, then your adaptive immunity will start to change. So that we can call like a little bit of evolution. And that can get passed to offspring, not necessarily within their genome, but think through like breast milk and things like that. So you can give offspring some immunity. So for innate, the big thing is that we say is not specific. Versus adaptive, we say specific. Oop. Why'd it go away? Okay. Hannah, you got anything else about innate or adaptive, either one?
I was just gonna say innate doesn't really take as much time to evolve and be there, like adaptive, and you have to take time. It takes time.
Okay, so innate is a faster response. That is totally correct. And this is longer response. We'll see a slide with that. Lakin, anything else you can think of?
No,
Pass,
pass.
passing on. Christina?
Innate does a lot of like protection, or it needs like a lot of protection.
Like, give me an example. Because I would say they both protect, but through different ways. Like, what's the first line of defense for innate?
Like skin.
Skin. Yeah. All right. So innate immunity involves many more parts of the body, whereas adaptive is really just going to be lymphocytes. And in that, it's going to be B cells and T cells. All right. Okay. So that was just some little different things about innate and adaptive. Obviously, this is your real big difference. So here we just talked about skin, right? So that's our first line of defense. But you think of all the epithelial throughout our body, especially if it's receiving information from the external environment, which is why you see lungs also have protection from their epithelial. And even for reproduction for females, the epithelium of the vagina is actually different for adults, and again, for protection purposes. Okay, when it comes to the things that are actually doing the work, so we've got a whole bunch of different cells versus that adaptive is really just the B cells and the T cells, and that's all. When we look at molecules, so really we're talking almost always about proteins. We've got RNases. Those cut or break down RNA. DNases cut and break down DNA. Proteases cut and break down protein. So if you've got a cell that's either infected or you've actually found a pathogen like a bacteria, any of these enzymes can work to try to break it down and lyse it so it no longer is functioning. We will talk a little bit about polysaccharides, but for the most part, when we think about polysaccharides, just think that you've got a cell, and on the outside of the cell are little tiny markers, polysaccharides that can tell. Could you actually see the little red thing I drew? Sorry. Can you see that up top? Nope. Let's change color. There we go. All right, cell. Still can't. How about there? Cell? Oh, there. There's a little tiny cell, and then just these little markers hanging off of it that tell the rest of the world, no, you still can't see it. This
Wait, you
thing!
drew it on the yellow.
Yes!
I thought you were drawing it on the grass.
Sorry. So any little marker outside the cell that tells the rest of the cells around the rest of the body what it is, those are often polysaccharides or little protein sugar markers or fat and sugar markers, but there's just things outside the cell. Okay, and we'll get into all these different types of things that protect the body.
Natural killer cell.
Yeah, they're there. They're there. All right, let's talk about the soluble proteins. And again, this is really all of those enzymes we talked about in those other pages. So first off, we know not all viruses are DNA viruses, which is why you not only have the DNA ACE, but that's also why you have RNA ACEs, because viruses can be either, and we don't want their DNA or RNA to stay around. In our tears and breast milk, both of those can have antibodies found within them. Even our saliva can have antibodies in it. So you can imagine anything that comes into our nose can get trapped by that mucus and then sometimes goes into our mouth. It's kind of gross, but nonetheless there's some antibodies there. Or you swallow it and it goes down to your stomach and it dies because of acid. And then we also have, in some of our cells, we have lysozymes. And those are enzymes that digest they digest things you bring from the outside. We pull it into the cell, and then we digest it into tiny little pieces. And then we also have antibacterial proteins. They're really helpful for killing bacteria. Not for killing viruses because they're antibacterial. All right, so in this slide, we're starting to see some of the different white blood cells. So if I can focus in on this. Okay, so we've got red blood cells, which is not a white blood cell. It is just the red blood cells, and they have nothing to do with immunity. Then we've got monocytes, and so what it really should have told us on that slide was that monocytes turn into macrophages. And macro means big, and phage means to eat. So these are big eaters. They are moving throughout the bloodstream, and they may get called into a certain area, which means they come out of the bloodstream and into a tissue and are there to eat things that shouldn't be there. Eosinophils, that's another type of white blood cells. It primarily protects against parasites. I'm going to run out of space. Against parasites. Then we have our lymphocytes. So from here, we're going to get natural killer cells. We're going to get B cells. And we're going to get T cells. Just to note, B cells and T cells are the adaptive immunity cells. Natural killer cells are innate. They are not considered part of the adaptive group. All right, platelets, those help blood clotting and have nothing to do with the immune system, other than we don't want to create a big open wound. We want to seal it up. And neutrophil is just another white blood cell, again, involved in attacking and killing different pathogens. Okay, so in this picture, this has to be a macrophage, and the reason I know it's a macrophage is because it is trying to eat these little bacteria things. So there's something on the macrophage that was able to recognize the bacteria, and when it does, it tries to engulf it. So it puts out its microtubules, which creates more of a foot. So that's why we see this pseudopod kind of growing. It grows towards the area of the infection. It puts an arm around it, and then you pull it in. So now it becomes this vesicle full of bacteria. And then this is where those lysozymes come into play. So in there are the lysozymes. And they are going to digest it and break it down to its itty-bitty pieces. And great. Then we've killed the bacteria. But there's another thing this cell might do, which it does not note in the book, but it also may become an antigen presenting cell. Okay, so when I explain antigen presenting cells, I can't do it in here. We have no markers in here. That sucks. All right. Does anyone? Okay, this is. I don't have.
I think if you open up the cabinet down there, there's some.
How do you know these secrets?
I come in here a lot.
Which cabinet, though?
The bottom one with like the computer stuff. I think they're like off to the left side.
Oh my goodness. Sam knows the secret stash. All right, so here's the deal. This is a bacteria, to those that are online and can't see me, I am holding an expo marker. And the expo marker has the marker piece and then it has the top. If this was the bacteria and you're a macrophage, you just ate the bacteria, gobbled it up, and broke it down into a bunch of pieces. But what you want to do as an antigen presenting cell is tell the rest of the cells nearby, there's bacteria here. I'm going to show you just a piece of what it looks like. And so then it cuts off, right, already gobbled up, and it presents to the outside world this tiny piece of the full bacteria and shows the rest of the cells nearby, look out for something that looks like this, and when you see it, go gobble it up and kill it. So they're very helpful in telling the rest of the cells what to look out for. So that's an antigen-presenting cell. Okay, and then the other thing we're seeing here, this thing is a white blood cell, and those are the pseudopods going up and around and trying to eat this green thing, which is the bacteria. There are tons of videos out there that show bacteria being eaten by macrophages. Okay, let's go. We're not going. All right, so I said we talked about neutrophils, and they're another type of white blood cell, and they primarily are traveling through the bloodstream, and something has to tell them to leave this bloodstream. So why? Oh, that's a worse color there. Show up here. How about down here? Can you read that? Yep. Why does a neutrophil leave the bloodstream? It doesn't just like randomly go along and go, oh, I'll just kind of leave the bloodstream right here and roam around in this tissue. It is attracted to a certain smell, and those things are called chemoattractants. So every chemical has its own signature smell in our body, and so white blood cells know what to go towards, what smell to go towards. The process of going towards a chemical is called chemotaxis. So in this figure, we've already left the bloodstream and you're in tissue. And how do I know that? That's because it's talking about its microtubules already. And they have started to arrange themselves and they're heading towards the bacteria. So they're heading towards the smell, and they will maybe eat it or send out a signal to some other type of cell to destroy that bacteria. If it was going to, so the steps before this is leaving the bloodstream, and for that process, it's very similar to a cancer cell being metastatic, breaking through the blood vessel. So it's got to hang on to the blood vessel first, break through the blood vessel, and then weave its way into the tissue. That's the almost identical pathway, well, reversed, for something that's in the tissue, cancer cell, and it breaks through the tissue and breaks its way into the bloodstream and then travels to another place. So this is the example of how does a cancer cell get abilities to move? Well, because white blood cells already had that ability, and so it's within our genome for them to have that information. Okay, so that's chemotaxis, going towards the smell of something. All right, so how now do those innate cells kill? We already talked about phagocytosis, though there might be another slide about it. But you need to understand how complement proteins work in a very basic sense, because they can get really involved, and then how natural killer cells work. All right, these are complement proteins. So what's the idea of how this works? This whole thing is a bacteria. Okay, and what we're seeing, the image is actually showing you the membrane or the wall of the bacteria or whatever pathogen is roaming around. So these, you see C7, C6, we don't need to memorize them, but those C's stand for complement proteins. Here's kind of how I remember complement proteins and how they function. If you meet a new person and they give you a compliment, and every day you see them in the morning, they give you a compliment, you're probably going to want to hang out around them because they make you feel better about yourself. So complement proteins like to hang out with each other. And then let's take it the next step. So you got a lot of friends that are all complimenting each other, and you join your own group, and then you circle up and you hold hands as a group. Okay, so now you've got all these complement proteins in a circle holding hands, and you land on a bacteria, and then you start stomping your feet, and you make a hole. Literally, this is what they do. They land on bacteria as a group, start creating holes into bacteria or other pathogens, and then once the hole's there, all the water in the interstitial fluid comes flying in, and it makes this bacteria explode. So that is one method how a innate system, because these are neutrophils or other white blood cells that are doing this. You get complement proteins that are floating around in the blood. They see that bacteria, and they join in a circle, holding hands, start stomping, create the hole, water gushes in, bacteria or other pathogen just explodes. All right, one nice, easy, cool way of how it works. There are other functions for complement proteins, but for the sake of this class, we're going to keep it just to that one. Create giant pores, let water gush in. Okay, natural killer cells. Paige, take it over.
Okay, all right, so,
I thought you were really going to. Probably
She
not.
could.
I know.
So natural killer cells, again, the innate system. Although they come from lymphocytes, they are part of the innate system. And they can kill in a couple different ways, but one of the main ways is, so here's a natural killer cell. This is the whole thing. And it is landing on top of a virus or a tumor cell, because they can work on both. Their initial job is tumors. Or viruses. That's how we figured out what they do, but then we also realized they noticed when our own cells became tumor cells. All right, so the best way for them to do their job, similar to complement proteins, but this is just the cell that does it, also creates a giant pore. So it creates a pore, okay, and then it releases these little digestive granules into the virus and it kills it. So again, some type of lysozyme or some type of digestive enzyme gets released into the tumor or the virus, and that's how they die. The other way might be by recognizing that this cell has FAS or the trail receptor out and about, which means if I lock up with it, I can activate apoptosis. So if you can create this setup, which should only happen when something is a virus or a tumor or a cell that needs to die, you won't see this setup on a healthy cell. They won't have the FAS receptor up there, the trail receptor showing. But once we show that, then the natural killer cell come in, docks, and again can cause apoptosis or some other type of cell death. And then the third way, so we go 1, 2, and then the third way is just it's going to release more signals. So cytokines are signaling proteins of the immune system. Okay, and it's just going to tell that signal is going to be received by other cells, and it's going to help bring in more proteins to kill off the virus or the tumor. All right, so if you don't know what these mean, these are interferons. And this is tumor necrosis factor. So it kind of depends what it came across. Interferons are used to fight viruses. And obviously tumor necrosis factor is there to fight tumors. Okay, so depending on what it runs into, it will release those cytokines to pull in more cells to help. Anything you'd like to add, Paige?
no, I think we covered it all.
Good, all right. All right, so here we're just seeing how is it that the natural killer cells get the signal to kill. There should always be a fail-safe because you never want your immune system just going out and killing anything it runs into. So here is the fail-safe for natural killer cells to either kill or not kill. Again, on the outside of all our cells are little markers, so little things just hanging off. And here we've got a little green protein, and it will dock with this activated inhibitory receptor, meaning it is inhibiting the actions of the natural killer cell. You do not want a healthy cell hooking up to a natural killer cell and it activates the natural killer cell. We want it to inhibit. So it tells the natural killer cell, don't kill me. I've got the normal signal outside my cell, which means I'm healthy. Don't do anything. Now, if it comes across a cell that has either been infected or might be a tumor cell, well, then it's got a different marker on the outside. And so it is going to interact with a different protein from the natural killer cell, and that activates the natural killer cell. So in that system, now you're going to see maybe the release of lysozymes, or it's going to activate apoptosis, something like that. So that's its mechanism to control itself. Okay, moving to the lymphatic system and how the lymphatic system is different from innate. So the lymphatic system is generally considered part of the acquired. And you all have T cells and B cells that will reside in lymphatic tissue, meaning your lymph nodes, your tonsils, your spleen is going to play a big role. And then even within your digestive tract, there's little patches of lymphatic tissue that check to see if there's anything bad in what you just ate. You didn't wash your apple or something like that. It brings in here the thymus, because that's going to be useful for T cells. T cells will mature in the thymus. And then it points out bone marrow, because all hemopoietic, oh, there's no way I'm going to spell this right. All blood stem cells are in the bone. So whether you're a white blood cell, neutrophil, T cell, B cell, eosinophil, or a red blood cell, or a megakaryocyte that makes platelets, they're all first start out in our bone, and then they may move, if it's a T cell, up to the thymus to get challenged. And I'll show you what that challenge is in a little bit. But nonetheless, lymphatic system has all your B and T cells. They will start in one place and then move and land in the lymph tissue and just hang out there. Okay, let's see, who do I want to ask? Who are you going to challenge? Let's challenge Brooke. Oh, no, I forgot my question. Hold on, it'll come back. Nope, it's gone. Okay, lucky you. You passed. All right, so again, adaptive, immune system. We've got two different categories. We've got something called humoral, something called cellular. Humoral does not mean it's funny. It just means back in the day, they said there are 4 different liquids in the body. And so they were called the humors. So you've got blood and bile and I can't remember the other humors. Anyhow, so we're just talking about the blood, but really we're just talking about B cells. Okay, cellular is T cells. So humoral is going to have the antibodies, because antibodies will float through your bloodstream, and B cells make your antibodies. Cellular means cellular killing. It is going to directly kill cells. Humoral antibodies, humoral anything is not a direct killer. It just marks something for killing. An antibody will land on a pathogen, tag it, and go, kill this thing. That's part of humoral immunity. Sorry, this isn't showing up very well, is it? I'm going to turn off this side of the lights. Maybe that'll help. Better? Oh, words.
Oh yeah, that looks good.
All right, so cellular then means direct killing a cell. You're going to kill what other bad cell or pathogen you run into. And that is all done by T cells, but more specifically TC cells, which are cytotoxic, right? Toxic being bad, cyto meaning cell, so it's bad for the cells that run into these TC cells. Okay, so here comes how we're going to check to make sure T cells are ready to go. There's, like I said when we looked at natural killer cells, there's always a level of regulation. You never want your immune system to just start sending out cells if they're going to be overactive. So in the bone marrow, like we said, has all the stem cells. That's the word I was trying to write, hemopoietic. That is your stem cell that turns into white blood cells, red blood cells, and platelets. What we're seeing here is the making of either a B cell or the making of a T cell. But if it's going to be a T cell, first it's got to go to the thymus, and that's where it gets its name, thymus T cell. And in the thymus, it's going to be challenged to act against our own cells. You don't want it attacking your own cells. So if a T cell, baby T cell, goes to the thymus presents its own cell to the T cell, and the T cell goes and attacks it and kills it, well, then the body knows this particular little baby T cell that tried to mature actually attacks our own body, so we need to kill it off. We don't want it to continue to grow and go out there in the rest of your body and start killing your own cells. So it would be challenged. It failed the challenge, and we killed off its lineage. The B cell gets challenged within the bone marrow. So it doesn't need to go to another organ, but it will also get challenged to see if it recognizes and fights off its own body. If it does, then we'll just kill it out. Okay, and then so this is saying in this part, B and T cells have been challenged. They are now mature. They know what to go fight. So now they're going to hang out in the lymph organs, whether it be a lymph node, a tonsil, Peyer's patches in the stomach, anything like that. They'll just wait. Now I got my question back to Brooke. Why do we have cells sitting in lymphatic tissue to check for pathogens?
A lot of times, lymphatic tissue is the first place that things metastasize.
Yes, for cancer, but what if it's just like a regular virus or pathogen? Why hang out in the lymphatic tissue? Why not hang out in the skin or...
I just feel like that's easier release to everywhere.
Okay. You're not wrong, Caleb. Do you have any other additional information? All right, I don't think I'm phrasing my question right. So the reason they hang out in the lymphatic tissue is because all fluid at one point will get pulled into lymphatic tissue and sample. Because you have to retain the fluid within your body so that you don't dehydrate. When stuff goes through the bloodstream, it leaves the bloodstream, goes into the organs it's supposed to be working on, and then that extra fluid we need to pull back into our bloodstream, but it goes through the lymphatic system first. Okay, so lymphatic system will get basically all the fluid or sample all the fluid that is in your body. On to antibodies. So remember, these are made by B cells. There are 5 different types or classifications of antibodies. You can't see them on that screen. Jeez. Can you see IgA, IgD, IgE? I don't know if you can see that on the screen. But on your slides, it says IgA, IgD, E, G, and M. It stands for immunoglobulin A, D, E, G, M. So from this, that's 5, and we end up with millions of antibodies just by having these 5 different classifications. I don't need you to memorize the differences between those classifications. You just know that they exist. We will talk maybe a little bit more about IgG and IgM. Okay, but you should know the structure of an antibody. How we determine if it's A, D, E, G, or M is by looking at its constant region. So there's only 5 different types of constant regions. And then we've got this light chain, this other light chain on this side. And then we've got another heavy chain that links onto your constant region. So here's the heavy chain. And what we've created by having the light chain and the heavy chain come together is a spot that interacts with either a cell that is like a pathogen or anything else that shouldn't be there, and an antibody notices it and it binds within it. It does not bind in the middle. It binds on those sides. So there's 2 binding points for each antibody, and they're very specific. How that binding site looks is extraordinarily specific for that particular pathogen. All right, it's just showing you a little bit more on how this binding site is made to look in a very specific way that will only bind basically one pathogen. Its job is to notice that pathogen and kill off that pathogen. That's all I need you to know from that slide. All right, so how do we end up creating millions of different antibodies when you only have 5 different classifications? And that's all by looking at the DNA of a B cell. I mean, all our cells have this, but the B cells are what use it. And they will mix and match their variable and their constant regions to come out with new antibodies. So, I mean, it's like having a bag of Legos and telling everyone to make an antibody using the bag of Legos, and everyone's Lego is going to look different. So they're just rearranging your Legos in a different order, and you end up with a new antibody. So that's pretty cool how we can continually make new antibodies for different pathogens. All right, this again is, I've kind of already talked about this. This is where we're going to, how we know to make more of a certain antibody. So we're going to have a bunch of different B cells because each one of them is going to make one specific antibody. B1 only makes that little blue antibody. B2 only makes that green antibody. It should be 3, way to count there, book. Only makes the red antibody. Okay, so you're making all of these cells all the time, but they are, for the most part, just kind of dormant, just waiting to get called up. So they're sitting there in the lymphatic tissue. You know, something passes by and it goes, do I match with this? Is this a pathogen? Do I match it? And if it does, which we see in this B2 cell, this little antigen, a piece of the bacteria or whatever, was able to bind within that particular binding site. And what that acts to do is it just says, now make a whole bunch of these B2 cells because I see the pathogen. Here's what it looks like, and I'm the only cell that can make the antibody to fight that pathogen. So go make a whole bunch of me, and I'll release my antibodies to go get that pathogen. So this is clonal expansion. Clonal expansion is huge for B cells. We just expanded that one clone, that one B2 clone. This is kind of cool too. At the left-hand side, we've got a resting B cell. Like I said, your cells are all there. They're just hanging out waiting to see if their antibody works on a pathogen. So what you see in this is you've got this huge nucleus, and then you've got the rest of it. Versus the antibody that is, or the B cell that has now been clonally expanded, but we've only got one of them, but this one is active. This one is making antibodies. And how do you know that? Jillian. I think you're done being a bio major. We'll just pass you now. Nice job, right? So in this particular setup, Jillian told us that Antibodies are proteins. Where do we make proteins that are to leave the body? We make them on our rough ER. So for this cell, it's got to ramp up its rough ER so that it makes a ton of that particular antibody. Next level question, who do I want to tackle? All right, we'll go with Tyler. You ready? What other cell does this? What other cell has a nucleus and then matures, kicks out its nucleus, and then only has a whole bunch of rough ER because it needs to make one protein really well?
I was gonna say like viruses, but I don't think so.
You know what? But that's not a bad way to think about it, but it's not right, but I understand why you thought that. Anyone else want to take a shot at it? Yeah, you're smiling.
Red blood cells.
Red blood cells. And Emma, what's the protein that they're making? What do red blood cells need a ton of? They bind oxygen. It starts with an H. Hemoglobin, right? So any cell that has to make is really its job is to make proteins, like a ton of proteins. If they're active, you'll see that they're going to have a ton of rough ER. So for this one, that's just making a ton of that antibody. All right, so that's that. All right, so we've got two ways that an antibody can interact. Two different things. This first one, up top, we've got a bacterium that's pink, and then connected to it are a bunch of antibodies. So the antibodies were floating in your bloodstream or whatever, or maybe in the tissue. Saw this particular bacteria had the right binding site with the antibody, and so antibodies surrounded it. The process of doing this so that a macrophage will end up eating it is called opsonization. And it means yummy. So, putting the antibodies all around the bacteria make it yummy, and therefore a macrophage wants to go and eat it. And I did just think of something as a completely different note. So many of you did the podcast. You guys chose two different podcasts to do. Do you ever talk about what podcast you're doing? Because it's bizarre how some weeks you'll just all land almost on the same one. So a lot of you did a neuroendocrine tumor one, or you did the neuroendocrine one. It was childhood cancers, or it was males talking to someone about, was it penile cancer? I don't know. I don't remember. Anyhow, neuroendocrine tumors. One of you said that they'd never heard of neuroendocrine tumors before, and one of you said, I remember this in Vert Biz.
Oh, I was the one that said I didn't remember it.
And so, there's just for the Vert Biz folks, there's a slide that is on your exam always, and it goes primary, secondary, tertiary tumor. Remember that? And you tried to you had to figure out which one was the tumor and which one was. I don't know. You did well in that class. Maybe you didn't. Anyhow, so that was just a totally aside thing. I'm sorry. But I wanted to remark that most of you knew what neuroendocrine tumors were. All right, so we got opsonization. Antibodies locking onto bacteria, makes it yummy, macrophage sees it, and gobbles it up.
I wish people could see my notes for this class sometimes.
Why? Because I say really weird things.
Yummy.
Yummy. I should make opsonized cupcakes. Put little antibodies sticking off the cupcake, and then everyone. Oh, I need to do that for the next Phys class. Not for you guys. All right. Here's the next one. This one is when that. I'm going to go back a couple slides. Is more like this setup, where the natural killer cell is figuring out it should be on a certain cell based on what's on the outside of that cell. So here we have antibodies made the target, which called in natural killer cells. They're also going to verify should they be here with that activating ligand that we saw before, that activating, protein. But nonetheless, they will dock in, and then it will go and kill the target cell. All right, so two different ways. Make it yummy, macrophage comes in and get it, or mark it, and a natural killer cell will come in and kill it. All right. This is just explaining to you how the body responds to the first time you see like a virus, and then the second time you see a virus, and why we can say it's adaptive because of what happens. So,
Guys, pay attention. I think this is a test question.
no, this is a test question. Oh, wait, it might have been.
Yeah, it was this graph.
Was it? No. Hold on, you did see a graph, but that's not that.
Oh, the non-loss.
It's usually on a 406 exam. Ooh, but optimization is yummy. Okay, so we got to look at the bottom, and we got to look at the x and y. So the bottom is time, and the y-axis is the amount of antibody that you're making. So it's arbitrary units. One antibody is less than 10 antibodies, right? Okay, so you get that idea. The first time you're exposed to a pathogen for your adaptive immunity, it takes a little bit of time, one, to see it because you probably aren't making the proteins or the antibodies. But finally, you see it, you start producing antibodies. So like at day 10 is when you saw it. And then at day, 17, 18, we're maxing out with one antibody or 100,000 antibodies. But it's still less than, say, the next time we see it. So 40 days later, you're in class, somebody sneezes on you, and then within 10, less than, like this looks like 5-6 days, you spike up. So it is much faster. This was 10 days. This was like 5 days. And then look how much antibody you make. You make a ton more antibody. So our body basically adapted to this virus or whatever. The first time it saw it, okay, I see it. I'm going to make some antibodies. I'm going to store that memory. So this is memory at work. Next time it sees it, because it has a memory of it, it knows exactly what to do and can do it much faster and to a much larger extent. Okay, it is 10 o'clock, right? Yeah, so it's time for a break. So everyone gets like a 5-minute break to do whatever they want.